Carbon dioxide is transported from the tissues to the lungs via the blood. In this video, I am going to tell you how the transport of the carbon dioxide or in which form the blood transports the carbon dioxide. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you have subscribed my channel. You can also follow HM Learnings on Facebook, Instagram and Twitter. And today we are back with a video which is on the transport of carbon dioxide after a long time. So in this video we are actually going to in this part of this video we are actually going to be more concerned that how the carbon dioxide is being transported from the tissues to the lungs via the blood. What are the major forms by which the carbon dioxide can be transported, and apart from this, what is the contribution of the blood cell in transporting the carbon dioxide, and what is the contribution of the plasma in transporting the carbon dioxide. And then we will focus on the certain mechanisms like the chloride shift, and we will focus that what are the very important critical determinants of the carbon dioxide transport. In the next part of this video, we are going to concern with the Haldane effect, which is very important if you are talking about the carbon dioxide transport, and the carbon dioxide dissociation curve. The two very important concepts, so we will focus more on these two topics in the separate video. So today we are actually going to be more focused that what are the different forms in which the carbon dioxide can be transported. So you can see that it is being transported actually in the three forms. Dissolved form, it can be dissolved either in the plasma or it can be dissolved in the blood cells also. It can be transported in the form of a bicarbonate, and it can be transported in the form of the carboamino compounds. Now when we want to talk about the carbon dioxide, we tend to think more about the venous blood, the mixed venous blood, and not about the arterial blood. But it is very important that we need to think about the arterial blood as well as the mixed venous blood, because as you can see in this graph, the total carbon dioxide which is there in the arterial blood, A stands for the arterial blood, is around 48 milliliter per deciliter. That means 100 ml of the arterial blood is having 48 milliliters of the carbon dioxide. And if you see the mixed venous blood here, it is having a total carbon dioxide level of about 52 ml per deciliter of the venous blood. So not much gap is being there. About 4 ml per deciliter of the carbon dioxide is being added when the blood is passing from the artery to the vein. So this increment of about 4 ml of carbon dioxide in 100 ml of the blood is happening at the level of the capillaries where the gases exchange is happening. So now this transport mechanism means the blood or the various forms by which the blood is carrying the carbon dioxide has to compensate, has to take this increment of 4 ml per deciliter of the carbon dioxide from the tissues to the lungs to maintain the body in a steady state. So for that, what are the changes which bring about in these three forms to compensate that increment of 4 ml per deciliter of the carbon dioxide? So you can see, first we talk about the transport of the carbon dioxide via the arterial blood. So in that, if the total concentration of the carbon dioxide in the arterial blood is about 48 ml per deciliter, which accounts for 100%. In that 100%, the 5% is actually contributed by the form of the carbon dioxide, by the dissolved form of the carbon dioxide. This is both in the plasma as well as in the blood cells. Now in the bicarbonate, you can see 90% of the carbon dioxide is actually in the bicarbonate form in the arterial blood, and which accounts for about 43.2 ml per deciliter. And the remaining 5% is by the carboamino compounds. So what are carboamino compounds? The carbon dioxide combines with the free amino groups of the plasma proteins or the hemoglobin, form a bond called as a carboamino compound. So now the arterial blood actually is passing through the capillaries. At the capillaries there will be gases exchange, they will be adding up of the carbon dioxide. So that is now has changed from 48 to 52 mg per deciliter. So now what are the changes which are being bring about now, because the blood wants to, the same 100 ml of the blood now has to carry about 52 milliliters of the carbon dioxide. So now what happens, the dissolved level, the contribution by dissolving the carbon dioxide in the plasma or in the blood cell has been increased from 5% to 10%. The contribution of the bicarbonate is decreased, actually it is 69% now, and 21% is by the carbamino compound, a great increase in being there. So the dissolved and the carbamino compounds which are having limited role in transporting the carbon dioxide in the arterial blood has a major contribution in transporting the carbon dioxide in the venous blood. Okay, and that is how the body compensated for the production, the increment of the carbon dioxide, so that they can remove this carbon dioxide and maintain the body in a steady state. So this 4 ml per deciliter is being increased, and in that the 10% is being transported by the dissolved form, 69% is by the bicarbonate, and 21% is by the carbamino compounds. So now we focus on that, what is the contribution of the plasma and what is the contribution of the blood cell, particularly the RBC, in transporting the carbon dioxide. So in this we will talk about the plasma and the erythrocytes. So basically, if you are talking about 100% transport of the carbon dioxide, 11% is being transported by the plasma and 89% is actually transported via the action of the RBCs. So you can see the major role is basically the RBC which is playing. So as this is the tissue, this is the endothelial cell of the capillary, and this is a lumen of the capillary, this is RBC, and this is a lumen of the RBC. Okay, so the carbon dioxide concentration will be higher on the tissue side. So carbon dioxide will diffuse, passing the endothelial cell membrane, will enter first in the plasma. Now 11% of the entered carbon dioxide remain in the plasma, but majority of it, that is about 89%, enters the RBC membrane via the help of two gas channels, aquaporin 1 and the RH complex. Okay, so now this 11% will be divided into three forms as we have discussed in the previous slide: dissolved form, carbamino compound, and the bicarbonate form. So about 11 out of this 11%, 6% is in the dissolved form in the plasma. Less than 1% is there in a carbamino compound, which can form the carbon dioxide can form a carbamino compound with the plasma proteins, which is very, very less, almost insignificant, and 5% is actually combining with the water present in the plasma to form the carbonic acid, which can dissociate to form the bicarbonate plus H+. Now this process is extremely slow because, as you know, the reaction gets speed up when the enzyme is present, but there is no enzyme present here in the plasma to catalyze this reaction, to speed up this reaction. So as a result, the bicarbonate which is being formed is actually formed very slowly. So the contribution is very less, only 5%. So about 6 plus 5 is actually the 11%. The contribution by carbamino compound is very less. Now when we talk about the transport of the carbon dioxide by the RBC, 89% is being entered inside the RBC. After this, 4% is in a dissolved form and 64% is actually converted into the bicarbonate with the help of an enzyme called as a carbonic anhydrase. Now this enzyme is exclusively present in the RBC and not in the plasma, and that is why here the reaction catalyzing the conversion of the carbon dioxide into the bicarbonate is very rapid. It results in the formation of the bicarbonate, and this bicarbonate, because you want the reaction to move in the right direction, you want more and more bicarbonate formation, you want more and more carbon dioxide removal or transport. So the reaction should move in the right direction, and it can be removed in the right direction only when this bicarbonate is removed from the RBC into the plasma, so that the reaction can move in the right direction. So for that you have one ion channel on the RBC membrane, which is called as the ion exchanger 1, also called as a band-free protein. So this chloride bicarbonate exchanger will actually exchange one bicarbonate. It will send one bicarbonate out in exchange of the chloride. So the chloride enters inside the cell. This is called as a chloride shift. And what is the consequence of that? We will look into the next slide. So because we want the reaction to proceed in the right direction, we want more and more bicarbonate formation, we want more and more carbon dioxide transport. That is why we are actually flushing out the bicarbonate outside the RBC. So even though 64% of the bicarbonate is being found by the RBC, but majority of the bicarbonate you are going to find in the plasma only, because it is being actually sent out of the RBC into the plasma. Now, after this, the another thing which is very important in transport of the carbon dioxide is the carboamino compound. So here it is being contributed 21% in the transport of the carbon dioxide, and it is happening because of the reaction of the carbon dioxide with the amino group of the hemoglobin. So hemoglobin is actually a better protein which is allowing the amino group, which is allowing the carbon dioxide to bind to its amino group, because it is a better buffer. It is in the reaction of, if you can see here, in the reaction of the carboamino compound, one H plus is actually being lost, and because the deoxyhemoglobin is a buffer, is a better buffer than the plasma proteins, it can buff off that H plus and can proceed the reaction in the forward direction. So that is how more and more carboamino compounds can be found by the hemoglobin. Also, the concentration of the hemoglobin is actually higher than the plasma proteins, and it has many free amino groups. So that is why the contribution of the hemoglobin is more as compared to the plasma proteins. So by this you can understand that the carbon dioxide transport critically depends upon the carbonic anhydrase, which has a role of about transporting 64% of the carbon dioxide. Chloride shift, because it keeps this reaction to going on in the forward direction, and the hemoglobin, because it buffs off the extra H plus ions, so that here also the H plus ions which are forming in along with the bicarbonate can be buffed off, and the reaction can proceed in the forward direction. So these three things are very important in the carbon dioxide transport. And now about the chloride shift. So this you already know that the carbon dioxide will enter inside the cell, and in the presence of carbonic anhydrase it will form the carbonic acid, and then it will form the bicarbonate. Now, and this H plus will be buffered by the hemoglobin so that the reaction can proceed in the forward direction, and this bicarbonate will be exchanged with the chloride with the help of an ion channel called as the anion exchanger 1, also called as a band 3. And because this chloride enters inside the cell, and we know that the chloride is osmotically active, it actually leads to the diffusion of water, osmosis of water from its higher concentration to the lower concentration, leading to the swelling of the RBC, and that is why the hematocrit of the venous blood is actually 3% more than the arterial blood. So these reactions actually reversed at the lung site, and at the lung site what happens that this reaction is going to happen in the reverse direction, leading to release of the carbon dioxide. The chloride will also get released. As a result, the water will also diffuse out, and the RBC will come back to its original shape. So this was about the chloride shift. And then about the very important thing in the carbon dioxide transport is the carbon dioxide dissociation curve, which explains the Haldane effect, or the Haldane effect is explained by the carbon dioxide dissociation curve. So this we will do in a separate video, because this requires a lot of explanation and a lot of focus. So if you like this video, please like, share and comment. Let me know any video you want me to make, and please subscribe to HM Learnings. Till then, keep learning.